Gamma-glutamyl-transferase, or GGT, is an enzyme that plays a crucial role in the metabolism of glutathione, an important antioxidant in the body. GGT is found in the liver, specifically in the canaliculi of hepatic cells and the epithelial cells lining the biliary ductules. It's also present in the kidneys, pancreas, intestine, and many other tissues. However, GGT is not found in skeletal muscle tissue or bone. The clinical significance of GGT is quite extensive. Increased levels are observed in all hepatobiliary diseases, with levels increasing to 2 to 5 times the upper reference limit. This includes conditions like viral hepatitis and alcoholic cirrhosis, making it a very sensitive indicator for these conditions. Higher levels are also seen in intra- and posthepatic biliary tract obstruction, with levels increasing to 5 to 30 times the upper reference limit. Interestingly, GGT levels increase before and remain elevated longer than ALP, AST, and ALT. Additionally, GGT activity can be induced by drugs such as phenobarbital and phenytoin, as well as by alcohol consumption. When it comes to test methodology, GGT levels remain normal in the presence of bone disease and during pregnancy, which contrasts with alkaline phosphatase, where levels would be elevated. The reaction involves I-glutamyl p-nitroanilide and glycylglycine, which are colorless. It's important to note that hemolysis does not interfere with the test results. For reference ranges, the upper limit for males is 55 units per liter, while for females, it is 38 units per liter at 37 degrees Celsius. All right, let's talk about amylase, often abbreviated as AMS. You might be wondering where exactly amylase is found in the body. Well, it's located in the pancreas, salivary glands, small intestine, fallopian tubes, and several other tissues. Now let's dive into the test methodology. The test involves Y-glutamyl glycylglycine reacting with p-nitroanilane, which results in a yellow color. Increased serum levels in acute pancreatitis occur in 2 to 12 hours after the onset of pain, with peak values in 24 hours and return to normal in 3 to 4 days. Increased levels can also be seen in conditions such as mumps, perforated peptic ulcer, intestinal obstruction, cholecystitis, ruptured ectopic pregnancy, mesenteric infarction, and acute appendicitis. Lipase or LPS. Chromogenic methods measure the formation of soluble starch fragments. Tissue location found in the pancreas, with lesser amounts in gastric mucosa, intestinal mucosa, and adipose tissue. Clinical significance. Tissue location found in the pancreas with lesser amounts in gastric mucosa, intestinal mucosa, and adipose tissue. Increased serum levels in acute pancreatitis occur in 4 to 8 hours after the onset of pain, with peak values in 24 hours, and return to normal in 8 to 14 days. Increased levels are also seen in conditions such as perforated peptic ulcer, duodenal ulcers, intestinal obstruction, and cholecystitis. Test methodology: oleic acid emulsion 0.8% plus H2O. Cholinesterase. Sources of error: hemolysis, because hemoglobin inhibits LPS activity. Two related enzymes, acetylcholinesterase, also known as H or true cholinesterase, and acylcholine acylhydrolase, also referred to as PCG or pseudocholinesterase. True cholinesterase is found in red blood cells, lungs, spleen, nerve endings, and the gray matter of the brain. On the other hand, pseudocholinesterase is located in the liver, pancreas, heart, white matter of the brain, and serum. Pseudocholinesterase is found in serum in decreased amounts in hepatocellular disease due to decreased synthesis, for example, in conditions like hepatitis and cirrhosis. Decreased levels of PCAE also occur in cases of insecticide poisonings. Additionally, PCE testing can identify individuals with atypical forms who are at risk. Test methodology involves several considerations. One key source of error is hemolysis. Additionally, the presence of fatty acids can affect the results, as they are measured by decreased turbidity. It's important to carefully monitor these factors to ensure accurate measurements of PCG. Glucose 6-phosphate dehydrogenase, or G6PD, is an enzyme that plays a critical role in cellular metabolism. Tissue location: found in erythrocytes, adrenal glands, thymus, lymph nodes, and spleen. Decreased levels of G6PD are primarily significant in cases of deficiency, which is inherited as a sex-linked trait on the X chromosome. In G6PD deficiency, a drug-induced hemolytic anemia can occur when an individual is administered anti-malarial drugs or primaquine. Hemolysis may also be caused by infections and after ingestion of fava beans. Increased levels of G6PD are seen in megaloblastic anemias and acute myocardial infarction. Test methodology involves the reaction of glucose 6-phosphate with NADP to produce 6-phosphogluconate and NADPH. Decreased levels of G6PD require the analysis of a red blood cell hemolysate. Analysis of G6PD elevations requires a serum sample. The reference range for G6PD in red blood cells is 8 to 14 units per gram of hemoglobin.